Hello, everyone. In this second part of chapter 2 of the cell, we are going to dive into cell division. We're going to dive into apoptosis, which is cell death. We're going to take a look at stem cells and microbiome. So, as we dive into this, the first stop we are going to make is going to be on cell division and cell death. Now, as we think of cell division, we can wonder, okay, why do cells need to divide? Well, cells need to divide because the body needs to grow, right? When you consider the fertilization of an egg by a sperm cell, that initial cell that we call the zygote is going to go from a bare cells, simple cell, to a complex structure that is an organism, right? That process is the process of development when you consider the first cell and the fetus in its 9 months. You can never imagine that first cell, the zygote, it's what became the fetus in its 9th month. They don't look alike, right? Which gives the impression that it's completely some new organism, right? That is basically what we call development. Cell division is essential for maintaining the body in good health, right? To heal whenever there is an injury, right? And this process has to be tightly controlled. Now, cell division is also called mitosis. Mitosis, it's basically the process by which you're going from parent cell to two daughter cells, right? In this process, you have different stages, including what we call the interphase, the mitotic phase, and cytokinesis, which is actually the division of the cytoplasm. Now, let me digress. Let me digress here. Now, when we talk about cell division and we go back to the history of the study of cells and how they divide, biologists actually were focusing first on what is happening to DNA. When biologists looked at what was happening to DNA, they noticed that, well, the nucleus was able to split into two nuclei at some stage of the cell cycle. And they call that particular stage of the division of the nucleus mitosis. But then over time, they found that, wait a minute, not only the genetic information is a split during the cell cycle, but the cytoplasm also is distributed between the two new daughter cells. And that division of the cytoplasm accounts for something. And that something is what we call cytokinesis, right? So cell division allows not only to distribute the genetic information equally among two new daughter cells while maintaining integrity of the amount of chromosomes in the new daughter cells, but also each cell has to inherit every single aspect, qualitatively, right, of the cytoplasm. But we also know that during the development of an organism, we have some cells that were there, and then the cells at some point they disappear. For instance, when you consider this individual here, now when you consider the digits, the fingers in your hands, these fingers, you have 5 fingers. Now, when we go back in the embryonic development and the fetal development, we notice that during what we call organogenesis, the formation of organs, in the hands, you have a web here between fingers. Now, at some point, that web disappears, and then the fingers exist as individual units. In some instances, such as in this child here, that web didn't disappear. As consequence of that, we have two fused fingers in one, right? So then the question comes, okay, well, what does it take for that web between fingers to disappear? Well, this is when we come and talk about apoptosis. What is apoptosis? Apoptosis is what we call programmed cell death. What is programmed cell death? Well, remember, during the fetal development, right? The digits, the fingers, are connected to each other. But when a baby is born, the fingers are not connected with each other. Question, what happens? The body recognizes that because of the function, because of the evolutionary functions of the hand, the web has to disappear. The web has to disappear. But how is the web going to disappear? Well, the way this is going to happen is that this has to happen in a controlled manner. That's why you need programmed cell death, so that only the cells that are in the web are going to die off, while the cells that make up the fingers don't die off, right? So now, mitosis is about cell multiplication. We go from one cell to two cells, and each of the two cells is going to divide into two cells, and each cell is going to divide again. Now, programmed cell death, on the other hand, it's going to be about destroying some of the cells, right? Like in the web case, right? You have extra cells that make up the web, and these cells have to die. And the body has to be very intentional in causing the death of these cells so that you can have your hand that is a tool that is going to be perfect and be able to perform the role for which that hand is designed to be. Now, the cell cycle, it's indeed a cycle because it's a sequence of events that cause the production of a new cell, actually two new daughter cells, and these two new daughter cells themselves are going to go through the same process again and again, and it goes on and on and on. So the cell cycle is the sequence of events associated with cell division. That sequence of event really involves two major stages / three. The first stage, it's actually what we call the interphase. The interphase lasts about 90% of the life of a cell that is designed to divide. Okay? And then you have 10% of the time of that cell is going to be spent in the mitosis phase and the cytokinesis phase, right? Now, when we look closely, we're going to see that the interphase, it's a phase that is divided into three subphases. We have the phase that we call G1 phase, another phase that we call S phase, another one that we call G2 phase. In G1 phase, the cell is actually doing what it is designed to do in the body. Okay? It's going to fulfill its function. That cell is going to grow. then the cell is going to receive a signal that tells it to actually start getting ready for multiplication. That signal is going to trigger the cell to enter what we call the S phase. That S phase is for the synthesis of DNA. So we have DNA synthesis in here. Now, after that DNA is synthesized, the cell has to make sure that that DNA is error-free, right? That control has to happen. And once that control happens, then the cell is going to enter a G2 phase. That G2 phase, it's a phase during which the cell is going to prepare all the material to actually perform a distribution of the genetic information. That's one. Two, a distribution of the cytoplasm so that each new daughter cell contains ribosomes, contain mitochondria, contain lysosomes, peroxisomes, the cytoskeleton, and so on and so forth, right? Now, after that G2 phase, then we go into the M phase. And in the M phase, we're going to have subphases, including prophase, metaphase, anaphase, and telophase. Telophase goes alongside with another phase that is cytokinesis, right? So, during the preparation of the cell cycle, well, we have the interphase, okay? During the interphase, DNA is going to be replicated, right? The cytoplasm is also going to be replicated, right? I already told you that you have a G1, S, and G2 phases in interphase. One thing I didn't mention, it's actually the G0 phase. Let's go back here. Now, some cells, when they divide, they get stuck in the G1 phase. In the G1 phase. And when they get stuck in the G1 phase, they actually just do their job, but never get worried about moving on to the S phase. This is true, for instance, for muscle cells. This is also true for nerve cells. And when the cells are in that very long G1 phase, we don't call that phase anymore G1. We call that phase G0 because the cell is stuck in terms of cell cycle, not necessarily in terms of function, right? They're still doing what they are designed to do in the body. However, they are not necessarily going through the specific process of the S phase and the G2 phase, right? So some cells get stuck in that G1 phase, and that G1 phase at that point is called G0, okay? Then we have DNA synthesis. That DNA synthesis, we have in the nucleus one chromosome that is going to replicate. So we have one copy and two copies of the same chromosome. The two copies are held together in a particular area right here. That particular area is called a centromere. Now, as the two chromosomes are held together, each other being a copy of each other, they are going to not be called chromosomes anymore. They're going to be called sister chromatids. In other words, when you hear in talking about the cell that we are mentioning sister chromatids, that means that the chromosomes have been replicated. They have gone through the S phase, right? Now you have two sister chromatids. And once the cell is in a stage where they have two sister chromatids, that means that the cells are relatively engaged to go through with cell division. Now, I have to mention, during interphase, the chromosomes, while they are replicated, they are not condensed yet. In other words, when you look at these cells under the microscope, you are not going to clearly see the chromosomes apart, right? The only time you're going to see the chromosomes apart, it's if these chromosomes start getting condensed, right? And when they start getting condensed, that's another story, right? This is when we can talk about prophase. In a prophase, the chromosome get condensed, right? Remember that chromosome, it's DNA and proteins called histones, right? And as these proteins and DNA are mingled, they form nucleosomes, and the nucleosome get compact. They form solenoids and so on. They become so compact that they are visible under a microscope, as you can see here. And when you can see them, then we say that the cell is in prophase. And at that stage of the prophase, the nuclear envelope disassembles. Also, the spindle that are ready and made up of microtubules are going to try now to target some areas of the chromosome that we call the centromeres, right? So the nuclear envelope is going to be disorganized. Then we have the metaphase. During metaphase, now the mitotic spindle right here, these microtubules you see here, this microtubule you see here are targeting the centromeres, and they are going to pull the centromeres toward opposite direction. So this is going to pull this direction, and this is going to pull this direction. So that because you have equal strength on both sides of the sister chromatids, they will end up lining up on the equator line, right? And this is what we call metaphase. Meta for middle, phase is the phase during which the chromosomes sit in the middle of the cell, right? Now, you have one set of centriole this way and another set of centriole this way. And the centrioles, they are made up of microtubules. And these very microtubules are responsible for the mitotic spindle that are nothing but microtubules. In other words, without the microtubules, you will not have the cell to go from prophase to metaphase, from metaphase to anaphase. That is not going to happen without the microtubules. Then we have anaphase. Anaphase is a stage during which the mitotic spindle keeps shrinking toward opposite direction and separate the two sister chromatids. And when they separate the two sister chromatids, now each of the sister chromatid is end up in one direction, while another one is end up in another direction. So at that point, the cell is way into separating the genetic information so that now you can have two future cells, right? And finally, you have the telophase. So during the telophase, finally you have the telophase. During the telophase, now notice that the chromosomes are now toward opposite direction, but you do see some membranes that are here. This is what we call the cleavage furrow, right? Cleavage furrow. That cleavage furrow tells you that these cells are now almost separated completely. You still have some reminiscent here, some remnant here of a previous structure that existed that was at one time one, and now we have two things, right? This is the nucleus that is forming here, another nucleus that is forming here. While this is happening during telophase, You have the formation of a new nucleus here, right? But also the distribution of the cytoplasm, so that you have some cytoplasm going here and some going here, okay? And again, this is the cleavage furrow, okay? So the spindle is going to disassemble, okay? The nuclear envelope reforms around each of the future nuclei. And cytokinesis, it's the division of the cytoplasm. I already mentioned this. And now for that division of the cytoplasm, microtubules play a very important role. Actin filaments play a very important role. I'm not going to bug you with the details, right? So organelles are going to distribute between the two daughter cells. The macromolecules are going to distribute between the two daughter cells. The cytoskeletal filaments are going to also distribute.